A randomized controlled trial, known as RCT, is a scientific, often medical experiment that aims to minimize bias when testing the effectiveness of a new treatment. The study consists of randomly assigning participants into an experimental group or controlled group where the outcome variable is reviewed. Steps to design a randomized controlled trial are taken in order to successfully produce the least most biased outcome. Step 1. Develop a hypothesis. According to Kendall 2003, analytical studies need a hypothesis that specifies an anticipated association between predictor and outcome variables so a statistically test of significance can be performed. It is essential that this is formulated prior to the beginning of the study. Without, what are the researchers actually studying, or what do they wish to prove? An example is drug A is more efficient in reducing the amount of cigarettes smoked by heavy smokers than drug B. Step 2. Defining inclusion and exclusion criteria. Poor sampling will undermine the generalizability of the study and potentially reduce the validity if sampling bias is introduced. Restrictions are applied to the population, and the inclusion and exclusion criteria should be carefully determined that creates a balance between generalizability and bias. To qualify for the study, participants must meet all requirements on the inclusion criteria. Those who don't fall under the exclusion criteria and cannot enter the study regardless of probable outcome of interest. According to Pawlik and Sosa, exclusion criteria serve to protect individuals who are at a high risk of developing adverse effects from the study intervention. Step 3. Randomization. Randomization allocates patients to either the experimental group or control group by chance. This method is to balance the known and unknown co-founding factors between the study groups, therefore reducing their impact on the relationship between the intervention and the outcomes observed. Those in the experimental group will receive the intervention, whereas those in the control group will receive the placebo. Step 4. Applying intervention and measuring outcome. It is important that the intervention is specifically defined and delivered consistently throughout the trial. For example, the timing and monitoring of the participants and their intervention doses, whether it be a drug, is vital to ensure it is clinically safe. To record the effectiveness of the intervention, an outcome, that is, a direct or indirect measurement of the participant's clinical or functional status, is documented. The outcome measurements will observe the difference between the experimental and control group in relation to those who received the intervention and those who did not. Step 5. Importance of blinding. A single-blind study is where participants are unaware of which group they have been allocated to, but the researchers do. A double-blinded study is that both researchers and participants are blind throughout the whole study. Blinding will reduce the likelihood of a biased outcome, as neither the researchers or participants have total control of the experiment. This can definitely be seen in the double-blind study. Step 6. Collecting data and quality control. When collecting data, special measures need to be considered, as randomization does not guarantee an equal distribution of patient characteristics between groups. For example, if there are 10 participants in each group, in the control group 2 participants may be smokers, whereas there could be 6 smokers in the intervention group. Data should be collected as continuous, rather than categorical or binary values. For example, Smoking status should be documented as an average per day, as this is more valuable than documenting if the patient smokes or not. This will also control the quality of the data which is produced throughout the study. Thank you for listening.